All right. So good morning and welcome everyone. For today's session, we have Dr. Stephanie King. Dr. King is a postdoctoral fellow in Dr. Galinda Med's lab within the neuroscience department at the University of Lethbridge. She earned her doctorate in biology from Washington State University under the advisement of Dr. Michael Skinner, one of the pioneers of inheritance, epigenetic transgenerational inheritance. Her background is in the transgenerational inheritance of metabolic changes following the exposure to environmental toxicants. Her current work in Med's lab looks at the epigenetic effects of transgenerational prenatal stress on the placenta and fetal cortex. So thank you so much, Stephanie, and we look forward to your talk.
Okay, thank you very much for the introduction, and thank you everybody for attending. I will be primarily discussing the environmental influences as well as epigenetic factors in multiple sclerosis. And so I'll go ahead and get started. Hopefully there will be some people pouring in as well, okay. So the etiology of multiple sclerosis remains somewhat elusive. However, there have been some studies that have revealed some indication of genetic susceptibility. Large-scale epidemiology studies of twins with MS have identified a concordance rate of about 25% in monozygotic or identical twins, whereas the concordance rate is only about 3% in dizygotic twins or non-twin siblings. If an individual has a first-degree relative with MS, then they have their risk of getting MS is about 30 times higher. And so what this indicates is that there is some level of genetic component in the etiology of the disease. And so genome-wide association studies have identified a predisposition associated with the major histocompatibility complex loci. Additionally, an assessment of almost 10,000 MS patients of European descent found more than 100 loci correlated to an increased MS risk. While many of these were related to MHC, about 30% of the loci identified were also found in genes in various immunological pathways, especially helper T cell differentiation, and other gene ontology pathways identified included cytokine pathways and signal transduction. So very interestingly, there was very little overlap in identified susceptibility loci between MS and other neurodegenerative disorders, including Alzheimer's and Parkinson's disease. This may suggest that the genetic etiology of MS could be initially driven by immunological factors. And so while we have discussed how there appears to be some level of genetic inheritable susceptibility in MS, genetic variation can only really account for about 30% of the overall MS heritability. And this suggests that the etiology of MS is likely to include environmental and lifestyle factors as well. And so several of these factors have been investigated for increasing the susceptibility to MS and exacerbating disease progression. The most well-studied include prior Epstein-Barr infection, cigarette smoking, low sun exposure and vitamin D levels, a history of childhood or adolescent obesity, as well as chronic stress. And more recently, other environmental exposures have been investigated as well. Areas with high use of pesticides have been shown to have an increased incidence of MS. Agricultural workers who are exposed to pesticides have an increased risk to develop MS, and this is particularly true for women agricultural workers. Exposure to sulfur dioxide air pollution has been tied to pediatric MS. Heavy metals like mercury have been linked to the development of MS due to the potential to accumulate in several glial cells, including oligodendrocytes. It should be noted that many of these correlations to MS risk in the third bullet are more novel and do need further investigation. And so how exactly can the environment impact biology? And so the phenomenon where genetic variation doesn't account for the majority of disease incidence isn't really unique to MS. In fact, the grand majority of diseases in the general population, like cardiovascular, metabolic, and neurodegenerative diseases, are affected more by environmental factors rather than genetic variability. Now, in the late 80s, David Barker and colleagues made one of the first observations that early life environmental insults can impact disease later on in life. And so infants born that were small for gestational age have an increased susceptibility to cardiovascular disease and metabolic dysfunction. And so following extensive further investigations, researchers proposed a concept called the developmental origins of health and disease. And so this hypothesis stated that the tissues and organs in sensitive periods of development would show permanent functional changes because of the adverse environment. These changes might significantly increase the susceptibility to a variety of chronic diseases in the individual, such as metabolic syndrome, fatty liver, neuropsychiatric disorders, etc. And so the developmental processes of these diseases are often accompanied by inflammatory and immune changes. And so an organism can respond to the surrounding environment during cell differentiation, and this changes the phenotype and gene expression without modifying the genetic code. And so this is known as developmental reprogramming. And so what exactly is epigenetics? And so developmental reprogramming and cellular plasticity involves epigenetics. This term literally means above or around genetics. It involves molecular factors or processes around the DNA that regulate genomic activity independent of the DNA sequence itself. What this allows for is rapid adaptation to a changing environment and ideally increases an organism's fitness to this new environment. And there are 5 known epigenetic mechanisms of gene regulation. We have DNA methylation, histone modification, non-coding RNAs, chromatin structure, and the newest identified epigenetic mechanism involves RNA modification. So for example, like RNA methylation. I will only go over the first 3 as they are the more classic epigenetic mechanisms within the literature. And so DNA methylation is one of the most well-characterized forms of epigenetic regulation, and it's associated with altered transcriptional activity of the associated gene. Molecularly, it involves adding a methyl group to a CpG dinucleotide residue. So in other words, a cytosine phosphate guanine. So that's what CpG means. DNA methylation is generally stable and enduring in cells. And so areas of this genome can be either hypermethylated or hypomethylated. Hypermethylation typically increases or decreases rather the expression of coding or non-coding areas of DNA, whereas hypomethylation tends to increase expression. Then we have histone modifications. And so these occur on the lysine residues of histone tails. Different modifications include acetylation, methylation, ubiquination, phosphorylation, and stimulation. These modifications control how either tightly wound or loose DNA is around the histones by either strengthening or weakening the bonds between the histone and the DNA itself. And so therefore, this controls how accessible or inaccessible genes are for transcription. And so histone acetylation leads to active transcription generally, while hypoacetylation can activate transcription. Histone modification of the H3 protein, a type of histone, is the most well-characterized form. It is responsible for the formation of what's known as heterochromatin. This is densely packed chromatin that is highly inaccessible. And so one of the most well-characterized examples of this is X chromosome inactivation. It's also known as the formation of a bar body. And so this is used for the transcriptional repression of certain areas of the genome, like for example, oncogenes, where those areas, if they were expressed, it would be harmful to the organism's physiology. And then we have non-coding RNAs, which are a very diverse epigenetic regulatory family. And these include long non-coding RNAs, small interfering RNAs, microRNAs, pRNAs, small nucleolar RNAs, as well as transfer RNAs. And I know that that sounds like a lot. They seem to be discovering more categories roughly every 3 to 5 years or so. And so this is a widely expanding group. These RNA types are considered epigenetic because they regulate post-transcriptional modification, gene expression, and the preservation or destruction of RNA transcripts. For our purposes, I'll mostly focus on microRNAs, which are a class of small non-coding RNAs. They're roughly about 21 to 25 nucleotides in length. They function in microRNA cleavage as well as transcriptional repression. And so changes in the microRNA expression are starting to be used as clinical biomarkers of disease, which we'll touch on a bit later. So the environment can induce changes to these epigenetic factors that are mitotically stable. So in other words, if a cell undergoes mitosis, these epigenetic changes continue to be present in the subsequent cells. These epigenetic changes can lead to altered expression and thus an altered transcriptome. And then they can affect physiology of an organism and potentially induce abnormalities as well as phenotypic variation. Unfortunately, in many cases, these changes in physiology can also induce a disease state. And so one can really notice the effects of genetics versus environmental and epigenetic influence in identical twins. And so large epidemiology studies of twins have identified specific traits or disease states that are more influenced by genetics like height and reading disabilities. Those that are represented, and so these are represented further up on the chart where we have greater genetic influence. Down at the very bottom, we have lesser or greater environmental influence. And so traits or diseases that have greater environmental influence or epigenetic influence are like, for example, rheumatoid arthritis as well as multiple sclerosis. And so roughly there is about a 25 concordance rate in identical twins, whereas fraternal twins, you'll only see around 3% of concordance rate. And so what's going on with the other 75% of identical twins where one gets MS and the other doesn't? One potential is the concept of epigenetic drift. Epigenetics has been shown to have a role in diseases from autoimmune diseases to cancer. It's also involved in some congenital diseases, endocrine, as well as neuropsychiatric disorders. And so one of the most well-studied examples of epigenetic drift involves DNA methylation. And so this is the epigenome of the chromosome 3 in a 3-year-old twins, as well as 50-year-old twins. So yellow represents where the chromosome is epigenetically similar, whereas the green and red shows where they are different. Now at 3 years of age, the twins are very similar. But as people age and they have different environmental exposures, the epigenome of twins becomes dramatically different. And so to present a particularly dramatic example of how environmental influences or epigenetics can influence the phenotype, I would like you to meet Susan and Jean. They are identical twins and 61 years old, but they look around 10 years apart in age. Susan, on the right, smoked for about 16 years of her life and sunbathes regularly, whereas Jean, who is on the left, avoided sun exposure and was never a smoker. Now, both smoking and UV light exposure have been shown to induce epigenetic and, in more severe cases, sometimes genetic changes that can affect both skin pigmentation patterns as well as skin elasticity to affect outward appearance, but it also induces disease states in internal systems as well. And so in the example of the twins, the exposure to smoking and UV light occurred later on in life, likely starting in adolescence or potentially adulthood, where most organs and tissues have completed growth and development. However, the earlier the exposure to an adverse environmental insult, the more an individual can actually be affected. So during development, dynamic changes in epigenetics can regulate the transcriptional activity of genes as cells acquire specific functions. Although select epigenetic marks are relatively malleable throughout development, a subset of epigenetic states is programmed early on and stable into adulthood. These epigenetic programs regulate essential cellular functions, including cell metabolism, survival, proliferation, and activity. Environmental dysregulation can lead to the inability to establish or maintain these essential stable epigenetic programs, and it has significant implications for long-term health outcomes. So over this next little bit, I will be going over some examples about how the environment or environmental exposures can impact epigenetic states, specifically with examples related to MS, like for example, vitamin D deficiency, smoking, and stress. And so an excellent example of how an early life exposure that it can increase susceptibility to MS is maternal vitamin D deficiency. So 2 studies from Finland and the United States collectively sampled about 36,000 mothers, and they identified a positive link between maternal vitamin D deficiency and risk of adult offspring developing multiple sclerosis. But what is the potential mechanism behind this correlation? Decreased availability of what's called maternal hydroxyvitamin D. It's the predominant but inactive form of vitamin D. That leads to decreased inactive vitamin D entering the placenta, and then it decreases the availability of the active metabolite required for genomic and non-genomic vitamin D receptor signaling within the fetus. Consequently, fetal vitamin D deficiency potentially disrupts the epigenetic programming during the development of target organs and tissues. So for example, like the brain, liver, and adipose tissue. Dysregulated epigenetic programming leads to long-term altered epigenetic states, which, as shown over here, can disrupt genomic accessibility of regulatory regions and downstream gene expression. So this can result in adverse long-term health outcomes in the offspring, including neurological and metabolic disorders. Now, vitamin D deficiency changes and its receptor, so that can alter DNA methylation, histone acetylation, and microRNA expression. Vitamin D receptors can regulate the expression of many genes by binding to their promoter and interacting with chromatin modifiers and remodelers. So note that in vitamin D in immune cells decreases immune cell proliferation and pro-inflammatory cytokine production, and it also increases the anti-inflammatory cytokine production. Within the nervous system, it increases the regulation of calcium uptake in the neurons, phagocytic activity of the microglia, the maturation of oligodendrocytes, and the activation of astrocytes. Epigenetic modification of these cell types following vitamin D deficiency may be a precursor to disease state by increasing neuroinflammation. And then there is a significant association between the duration of smoking and the progression from relapsing remitting MS to secondary progressive MS. And so this might have to do with the sheer level of toxicants present in tobacco, including cyanide and dioxins. So smoking has been shown to induce hypomethylation of the aryl-hydrocarbon receptor repressor. And so this in turn inhibits aryl-hydrocarbon receptor signaling, which leads to the enhancement of neuroinflammatory and neurodegeneration events. Sorry about my email. I would be awfully remiss if I didn't include some of my lab's work with this. Prenatal stress has been shown to increase the susceptibility of inflammatory and immune diseases. And patients with MS have reported that stress can trigger relapses and exacerbates clinical symptoms. As a result, the METS lab wanted to see if stress would alter epigenetic signaling in the animal MS model called experimental autoimmune encephalomyelitis, or also known as EAE. So they wanted to look for an experience-dependent biomarker of MS. in addition to investigate if chronic stress may exacerbate MS-like symptoms in this EAE model. So the EAE plus stress group had a more severe clinical symptoms like hind limb paralysis, as well as a loss of coordination when compared with the EAE group alone. So microRNA 146A and microRNA 155 are both biomarkers of human MS and have increased expression in active MS lesions. In the EAE model, these microRNAs appeared particularly responsive to stress and showed significant differences beyond changes related to EAE alone. Now, stress may therefore serve as an environmental epigenetic risk factor for disease severity in MS. And so there are certain epigenetic factors that I will go over in the development and progression of MS. And these include epigenetics in neuroinflammation, demyelination, as well as failure of remyelination. And so aberrant DNA methylation has been demonstrated in both neuronal tissues as well as peripheral blood leukocytes in MS patients. These methylation changes often occur prior to clinical detection. So Hune et al. Compared pathology-free areas of the brains from MS patients compared to pathology-free areas of controls. And so these were called normal appearing white matter, or NAWM. And so there were widespread DNA methylation changes across the genome, including over 900 different associated genes. To follow one gene, capsaicin Z, also known as CTSZ, that is a gene that is known to increase neuroinflammation by promoting the interleukin-1 beta Th17 axis. They identified hypomethylation of the gene that correlated directly to increased expression of that gene. And hypermethylated genes in this set tended to involve the actin filament-based processes in nucleotide metabolism, in addition to some oligodendrocyte-specific genes like myelin basic protein and SOX8. The hypomethylated genes tended to include lymphocyte-mediated immunity, as well as leukocyte-related pathways, which may relate to immune-mediated pathology in MS. And one specific mechanistic pathway, involving DNA methylation and demyelination has to do with the citrullinization of myelin basic protein. And so this is the conversion of arginine to citrullinine by the enzyme called peptidyl arginine deaminase. And so citrullinization of proteins can generate potential autoantigens. And so these contribute to T-cell autoaggression in the central nervous system. So increased levels of citrulinated myelin basic protein is observed in normal appearing white matter in MS patients, similar to what we saw to what they investigated in the last study. And so this may be due to a hypomethylation in the promoter of PAD2, in other words, the citrullinization enzyme gene. Hypomethylation of PAD2 correlated with an increase in expression of PAD2 in the peripheral blood mononuclear cells in MS patients. And so this mechanism is not really necessarily unique to MS. Citrullinization of specific proteins has been found to induce an immune response in rheumatoid arthritis, lupus, and Sjögren's syndrome as well. And so peripheral blood mononuclear cells are T cells, B cells, and natural killer cells. And so then we have remyelination failure. As mentioned, vitamin D availability is essential for oligodendrogenesis. And so in other words, the generation of oligodendrocytes. In the developing forebrain of mice, the entire oligodendrocyte population is generated from about 3 phases of oligodendrocyte proliferation and migration. After CNS development, the small fraction of oligodendrocyte a small fraction of oligodendrocyte progenitor cells remains undifferentiated. And so these adult oligodendrocyte progenitor cells are morphologically equivalent and they express the same markers as the oligodendrocyte progenitor cells present during development. However, they differ slightly from the developing oligodendrocyte progenitor cells in the fact that they differ in growth cell responsiveness, migration capacity, as well as cell cycle length. Now, having an environmental insult that can induce epigenetic changes during one of these three critical windows of development could actually change how a cell acts later on in its lineage. Now, under normal conditions, if one myelinating oligodendrocyte is lost, the adult oligodendrocyte progenitor cells are there as a replacement. So the oligodendrocyte progenitor cell migration and recruitments require several growth factors, including platelet-derived growth factor and the fibroblast growth factor. So both of these growth factors are significantly methylated in the normal appearing white matter of MS patients. Even pathological demyelinating plaques in MS still contain oligodendrocyte progenitor cells. However, these cells tend to fail to differentiate to replace the lost cells. Targeting epigenetic factors to influence oligodendrocyte progenitor cell differentiation as a means to promote myelin regeneration in MS is actually a really exciting potential therapeutic avenue. And at that last point of targeting specific epigenetic factors as a therapeutic in the future brings up a bigger question. I mentioned that epigenetic changes can be detected in peripheral blood leukocytes in individuals before they are clinically diagnosed with MS. So can the epigenome be used to identify individuals susceptible to MS before it actually starts? Furthermore, can we use the epigenome to not only predict MS, but use it for personalized medicine to treat or prevent MS before the clinical progression starts? I know that that sounds a little bit like a pipe dream, but that's one of the ultimate goals in the field of epigenetics, and some groups are actively working on trying to find a predictive biomarker. And so one promising study shows that differential patterns of DNA methylation is not only seen between MS patients and healthy controls, but also between MS patients in different stages of clinical disease, disease severity, and form of disease. So Kulikova et al. Performed a whole genome DNA methylation analysis of peripheral blood mononuclear cells in relapsing remitting MS and primary progressive MS patients. And then they compared them with healthy controls. A principal component analysis for data visualization demonstrated a clearly defined clustering of separate comparison groups compared with the controls. More differential DNA methylated regions were identified in primary progressive MS compared to relapsing remitting patients. Further refinement of these differential DNA methylated patterns may end up leading to a promising biomarker not only for MS risk, but also for the progression of disease. So in this talk, we have explored how vitamin D deficiency and as well as smoking and stress have been shown to induce epigenetic changes related to or exacerbating in MS. As well as we discussed how epigenetic factors can affect processes identified in pathology as well as the progression of MS, including neuroinflammation, demyelination, and remyelination failure. And so epigenetic marks appeared prior to the onset of MS, and they may lead to promising predictive and diagnostic biomarkers in the future. And so with that, I would like to thank my lab members, as well as CAHR, as well as NSERC for funding. Thank you so much.
Thank you, Stephanie. We have a few questions there. Looks like the whole box is full. So if you stop sharing the slide, you can go over the chat box.
Okay, let me try to see if I can, oh, it's up there. Okay, I thought I saw things popping up, but I wasn't quite sure.
We have first question from Salma.
Okay, yes, I see it. So it says, if genes can be modified by the environment due to epigenetics causing disease, is there any way for these modifications to be reversed as a cure? And that's actually a really great question. And so that, yes, actually that can occur. So a really good example for this is epigenetics inducing cancer. And so in some cases, cancer can actually stem not from genetic mutations, but actually from hypomethylation. And I kind of referred to this a little bit earlier, where you can have oncogenes that are not meant to be expressed. And so if those genes start to be expressed, that can induce cancer. So some cancer drugs are actually work to reverse certain epigenetic marks. And so they use that as a form of chemotherapy as opposed to other diseases. As far as therapeutics for MS, I'm not really necessarily sure if they're working on that. However, that would be a great question for clinicians. There is another question about how quickly or easily may the epigenetic effect of vitamin D deficiency in pregnancy be reversed if the mother or offsprings are subsequently administered vitamin D? Also another great question. I'm not necessarily sure about how quickly or easily that may be reversed. However, I think that especially if you hit that within pregnancy rather than waiting until they're neonatal. So for example, if you have vitamin D deficiency earlier on in pregnancy and potentially if you supplement that later on in pregnancy, it's entirely possible that those effects may be attenuated, just primarily because you have a lot of developmental plasticity still. However, if you try to wait until later on in life, for example, when all of the organs and tissues have already developed and they're kind of already set in their epigenetic state, then that becomes much more challenging. So in other words, the best practice is the earlier the intervention, the better off the patient outcome. And so another one said, similar question here, with the current technology, are we able to induce epigenetic changes such as controlled methylation of certain segments for therapy and prevention? So that is actually a really interesting question in the field, because I know that there have been some people out there that are looking into CRISPR-Cas9 technology specifically to reverse different methylation changes. So CRISPR-Cas9, if you're not familiar with it, is a technology that targets specific sequences, and they're trying to use that to reverse specific mutation states, but also inserting genes, that sort of thing. And so what they're looking into that with is effectively to insert the same sequence of DNA, but actually have it in a demethylated state or something similar. So I think that people are definitely looking into that, especially when it comes to highly targeted therapies for epigenetic states that induce disease. And so is there any data about how varying intensities of stress can differentially impact the EAE severity? Yes, I do believe that there is some data out there. Off of the top of my head, I am not entirely sure about the severity of stress. However, this is definitely something that a lot of MS patients express, is that when they are under high degrees of stress, something's going on with their life, that sort of thing, that they find that their symptoms are exacerbated during that period of time. However, this could change if you're looking at acute stress versus chronic stress. So in other words, chronic stress as in your life has been stressful for a long period of time, like you have a really heavy workload, versus acute stress, like for example, you were just in a car accident, that sort of thing. And so then we have another question regarding DNA methylation. Does hypermethylation or hypomethylation, so I think what you're asking is about, I think I see the follow-up question. Are there differences in MS severity between hypermethylation versus hypomethylation and DNA methylation? So what I think you're asking about is specifically as far as global hypomethylation, whereas you have more hypomethylation throughout the genome versus hypermethylation throughout the genome. I would say that the MS severity is likely more to do with what genes are hypermethylated versus what genes are hypomethylated. So hypomethylation typically results in the increased expression of genes, whereas hypermethylation tends to repress genes. So if you have hypomethylation of genes that induce immunological changes, so for example, genes that would induce T-cell aggression, proliferation, that sort of thing, then that would definitely exacerbate MS. Whereas if you have hypermethylation of the certain factors, like for example, the ones where we discussed remyelination failure. So if you have hypermethylation of certain factors like the FGF or something like that, that would induce those oligodendrocyte progenitor cells to remyelinate, then that would definitely cause exacerbation of MS severity. So I think I don't think it would be like overall hyper versus hypo, but I think it just has to do with the specific genes associated with that type of methylation. And so how useful would it be to find genetic biomarkers for MS if MS is not strictly a genetic-based disease? And so I think it's both. I think it's, so some people have a very strong lineage component in MS, and I think that that is very important to find genetic biomarkers to help them, especially if you can check for certain mutations that increase your susceptibility to MS. So for example, if you know that you have this susceptibility, then you can start treatments a lot sooner. You can monitor potential symptoms, that sort of thing. So for example, I have known a family where, you know, five members have actually been diagnosed with MS. So I think for those individuals, it's very important. So they have a strong lineage, whereas in other individuals, you know, it's not necessarily a genetic-based disease. So I think it's important to actually look at all components. And so I think it's important to look at both the epigenetic factors, but also the genetic factors as well. And so in my opinion, what is the most significant or impactful epigenetic factor for MS? That is actually really challenging to say because unfortunately, a lot of the studies only are looking at DNA methylation. Now, a lot of studies are also tend to be looking at different things like microRNAs, but DNA methylation tends to be the most well-characterized epigenetic mark. So it's the most popular to study because we have it very validated as something that affects disease. So I think it's actually very important to look at other factors as well that might just be kind of hidden causes. So for example, I haven't seen a lot of studies on histone modifications associated with MS. A lot of them just tend to be DNA methylation. So, so I'm not necessarily sure if DNA methylation is more impactful for MS or if it's just the most well studied in this regard. So another question was that you may have covered this, but I may have missed it. Can you tell us about epigenetic changes that are passed on from one generation to the next stably? Yeah, so that's actually part of my research, and that is a very complicated question. So a trait can actually be passed on to the next generation potentially. So under normal conditions, epigenetic marks tend to be wiped when it comes to the next generation. However, there are some very critical periods of time where the epigenome can actually be transmitted to the next generation. So with these exposures, they have to occur during germline development. And the reason why is that the germline is what is passed on to future generations. So if you modify the epigenetic code, so you reprogram the epigenetics within the germline, so either the sperm or the egg, those actual epigenetic changes have the potential to be passed on to future generations. So that is one of, and I would be happy to actually share some slides with you as well with that, but it is kind of more of a heavy topic that would take me about 15 minutes or so to fully explain, but I would be happy to do that in the future. So since melanin is known to influence vitamin D absorption, is there a known link between melanin levels and MS? You know, and that is a really interesting question. I am not sure about that one, unfortunately, but I would be happy to look it up. It is my understanding that that you tend to see more incidences of MS in northern populations. However, I'm not sure if that has anything necessarily to do with melanin levels. And so then we have another question. If these epigenetic changes target the HLA complex, what causes the disease progression into MS specifically and not any other autoimmune disease? So that is a good question. I wonder if it's because of certain things, like I discussed the citrullinization of certain proteins, and I kind of wonder if it's so much of a multifaceted disease that maybe you have to have certain things happening all at once. So for example, citrullinization of specific proteins has to do it basically creates an autoantigen potentially. And so I kind of wonder if that has to occur with certain proteins. So for example, if it in rheumatoid arthritis, for example, this occurs in fibrin and fibrinogen. And so that instead of the myelin basic protein that we see in MS. So I kind of wonder if that's part of the influence why you might have one autoimmune disease versus another, but then that's just one facet. But then the other aspect is that you would need to have that heightened level of immune response. Unfortunately, the etiology of a lot of these autoimmune diseases are really elusive. So I'm not really necessarily sure about that one. Let's see, any thoughts on why MS prevalence has been increasing other than increased and more accurate testing? So that could very well be due to increased environmental exposures. And so, for example, I was mentioning very early on in the presentation that some of the things that people are looking at is heavy metal exposure, increased exposure to air pollution specifically. So for example, sulfur dioxide is another one, as well as people that are exposed to certain high levels of pesticide also have that increased susceptibility. So that might, those type of things like, especially the heavy metal exposures might explain why we might have a increased level of MS compared to potentially about 100 years ago, but when we had less exposure to these types of environmental toxicants. And let's see. And I think we have one last question, and we might have more pop up, but are there known epigenetic changes between relapse and remission in MS? So that is a good question. I actually don't think that I have seen a study where they sample people in remission versus people when they have a relapse. You know, and I think that that could actually be very easily studied if you looked at the peripheral mononuclear blood cells. So that would actually be a really good idea for a study, and I would suggest potentially in putting a grant up for that, because that's a really good idea. And so then but to kind of more answer your question, there is a possibility that there could be, but that is a good idea. So then we have another one. Would very high levels of vitamin D during pregnancy have an effect on epigenetics? I would say probably yes. And the reason why is because vitamin D is actually fat soluble. So you can, in other words, it does, when you have kind of like an overdose of vitamin D, it doesn't clear the system. Like, for example, if you have very high levels of vitamin B, vitamin B, for example, that gets excreted through the urine. It's a water-soluble vitamin, so your body takes care of that and excretes that. Whereas certain fat-soluble vitamins, like, for example, vitamin A and vitamin D, if you have an overdose of those can actually cause a disease state. And so it's very important with fat-soluble vitamins that you have an actual, a good balance. And so you take the proper dosage. And so I would imagine that, yes, increased levels of vitamin D would probably also cause an adverse effect. However, I don't think that that has been studied, just primarily because overdoses of vitamin D are relatively rare. It's unfortunately the case where almost all of us are vitamin D deficient. But thank you so much for all of the questions. They were actually really well thought out.
Yeah, do we have more questions coming, or I'm not sure? Okay, so I really enjoyed today's session. It was less teaching, more of discussion. So thank you, Stephanie. Yeah, we really liked the talk.
Thank you so much for the opportunity. It was fun.
Yeah, thank you. And everybody else, we'll see you next Thursday.
Sounds good.
Bye.
Bye.